My name is Melissa Roe. I have this first project is extracting DNA. The second project is X inactivation. They both would be involved in the unit of genes or heredity. So probably do the X inactivation project first because it would follow really well after Mendel's laws. Because we'd be talking about Mendel's laws, how genes work, how things are inherited, and the funny thing about the tortoiseshells is that the females have two different, so they get two X chromosomes, so they can have a black colored or an orange color, and each cell determines whether the black or the orange is going to be activated, so that's what determines its significant coloring. So, it followed really well. And then for the DNA extraction, it would still work well with the same unit, but maybe a little bit further down the teaching, down the lessons, because we have to talk about the structure of DNA, how DNA works, what pieces are there, in order for the students to understand how detergent and alcohol can separate DNA from the rest of the material. So it's really important that they would understand the background information before we actually would get into the doing this learning center. But what's great is that the way I have it set up is it's a kind of a scaffolded lab experiment. So they'll be doing a lab write-up along with doing the experiment, and some pieces of the lab write-up are included in the board, and some pieces they have to do themselves. So, like, they have to come up with their own hypothesis. They'll have to write their own conclusion, but they won't have to write the background information. They won't have to do the research needed to do the experiment, because that's already included on the board. They just need to summarize it and understand how the information from the background or from the research, how does that reflect upon our experiment.